So we are live.
Sir, we are live.
Very good.
A warm good noon to everyone. I, Mrs. Rima Kuwar, Assistant Professor, Sir Dr. M.S. Gosavi, College of Pharmaceutical Education and Research, Nashik, feel honored to welcome you all to Day 5, Session 2 of 1 Week International e-FDP, e-STP on recent advancements in pharmaceuticals, academic research, ethics, and social perspective. It gives me great privilege to introduce today's second speaker, Session 2 speaker, that is Dr. Pursuhottam Dewang, founder, MD, and Chief Scientific Officer at Metajivan Agro Research Consortium in Bangalore, Karnataka. Sir has completed his PhD in organic chemistry. He is having total 21 years of post PhD academic and corporate experience. He is a member of Advisory Board Acharya and BM Reddy College of Pharmacy, Bangalore. He is also co-inventor of 3 clinical candidates, submitted IND applications and got accepted by US FDA successfully for clinical trials. He worked with USA-based biotech on drug discovery projects and co-inventor in patents. He is having more than 10 years of research experience. He is well versed with CRO model. Regarding his active contribution, he in association with Into Incorporation, now called as SK Chemicals of South Korea, and Eva Women's University, he executed the medicinal chemistry project to develop of backup compound to a preclinical candidate IN1130 and ALK5 inhibitor. He is expertise in designing, driving, and monitoring collaborative multidisciplinary research projects to develop novel products. He is also expertise in designing and developing contract research partnership, identifying business opportunities for research collaborations, developing business models, developing collaborative research proposal by working with multidisciplinary teams within the organization and across organization with team members of client's company. Regarding the awards and achievements of Sir has received Senior Research Fellowship from Council of Scientific and Industrial Research, New Delhi. He is nominated by Honorable Governor of Maharashtra as an expert on the Board of Research of Kavitri Bahinabai, North Maharashtra University, Jalgaon. Sir has 5 patents, 25 journal and 2 book contributions, 3 research or report publications in his account. The topic of today's session is Target, Engage and Destroy Protein of Interest, New Generation Therapy and PROTACs. Now I kindly request Sir to start your session. Please, Sir.
Thank you very much. So, thank you very much for the kind introduction. And is it visible? The slide is visible?
Yes, sir. Slides are visible.
Slides are visible. So, okay. I'm going ahead with this one. And as mentioned that today we are going to discuss, target, engage and degrade the protein of interest, new generation therapy, the PROTACs. Basically, the title itself clears you that this is regarding the targeting the and degrading the proteins of interest. The proteins of interest are basically the proteins that are involved into the disease generation. Okay, so we will see that one into the coming slides. So I will move on to the next slide where there is a brief introduction of mine, but as it has been discussed, I will keep this slide. But here, what I want to emphasize to you is that having an fortunate to have an experience from conceptualizing the drug discovery project up to the IND filing to the US FDA and get it approved, that kind of experience we are having today. So you are free to ask within this domain any questions that you have into the chat box or to directly to me. So I'll move on to the next slide where we will start discussing our topic. My question to all of you is that we are discussing today the PROTAC, but is the PROTAC really a $1 billion topic? Is it a really important topic in a drug discovery today? Is it the medicinal chemistry are across the globe are looking towards this approach as a solution for them to discover the new drugs? So this slide will give you a clear understanding what is happening. See, when the companies put their hard-earned monies, or anybody puts their hard-earned monies into the discovery or research or in a production, that means that field is thriving. If you can see that the first graph onto the right hand side, that the growth of this field, it shows the funding and investment cumulative amount invested by year. So you can see that almost billions of dollars are being invested since 2014. It was just 264, but um, you can see that the US 5,326 millions means 5 billion plus dollars, and this accounts into the Indian context almost around 41,000 crore rupees. So my emphasis to is that today's topic is very, very hot topic, and the world is more and more inclining towards implementing these strategies for the drug discovery. Funding and investment numbers also will show you that more than USD 2.3 billion has been raised by the companies using this kind of a research. So those who are listening to me today, they should understand that uh, if you are into the drug discovery and you don't have the clear understanding of the PROTAC subject, you should try to understand it. You should try to implement it. Millions or dollars, I mean, actually the number is in billions of dollars are being raised. Onto the third figure, onto the right, onto the extreme right of this slide shows that which are the companies and how much funding they have raised into the recent years. So you can clearly see that Radius, Arvinas, and all those companies are at the top of the graph, and they have raised a couple of millions into the couple of millions of funding using this topic. So now what is this topic exactly, that we will understand. So here in a body there is a mechanism to degrade the unnecessary or those who have become unnecessary proteins. Okay, and that mechanism is called one of the mechanism is called ubiquitin dependent proteolysis system. Please remember this word because we will be using this word throughout our discussions couple of times. This is ubiquitin dependent proteolysis system, and in short let us call this system as a UPS. It is now widely appreciated that UPS plays a critical role in regulating a wide variety of cellular pathways, including cell growth and proliferation, apoptosis, protein quality control, DNA repair, transcription, and immune response. With a few exceptions only, destruction of cellular proteins by proteasome is gated by their ubiquitination. So that ubiquitination is a key aspect that we are going to do. Covalent modification of a ubiquitin chains act as a sort of a tag signaling their proteolysis. That means when the body wants to degrade some protein and doesn't want that to be into the body tags that particular protein with the ubiquitin. The UPS, as the short form for the ubiquitin dependent proteolysis system, conserve throughout the eukaryotic cells, employs the a common regulated enzyme cascade consisting minimally of E1, E2, and E3 activities. You can see into the figure here. In a brief, ubiquitin activation enzyme E1 uses the energy from the ATP hydrolysis to generate the high energy thioester bond between the carboxyl terminal glycine residue of ubiquitin and catalytic cysteine residue within the E1 itself. So this is the first step. This activated ubiquitin is then transferred to an active site cysteine residue within one of the several dozens of ubiquitin conjugated enzymes E2. You can see the second step in the figure. Finally, the charged E2 enzyme cooperates with one of the hundreds of ubiquitin ligase E3 to transfer the activated ubiquitin to amino group of the lysine residue within the target protein. In most cases studied thus far, this initial ubiquitin serve as an acceptor for further cycles of ubiquitin modification. That means this goes into the cycle and multiple ubiquitin modifications are take place. Subsequent ubiquitin addition and can occur through the isopeptide linkage on all of the ubiquitin 7 lysine residues as well as the amino terminal primary amine therapy chains. So what happens after this ubiquitin is activated and conjugated to a target protein by the conserved series of E3, E2 and E1, E1, E2 and E3 ubiquitin ligase activities. Um, Polyubiquitination proteins are recruited via ubiquitin receptor to 20S6 proteasome multi-subunit barrel-shaped cellular protease, and you could see that substrate and ubiquitin, multiple ubiquitins are here, and that finally leads to the proteolysis or the degradation of that substrate. So what is happening here, if you understand that, here for most important for you to understand is that only that ubiquitin or this pathway is particularly there into the body, but what the scientists are doing smartly, we will see into the next slide, that this mechanism, you only need to understand that there is a ubiquitin and which is being recruited to the protein of interest by the E3 ligase enzyme. This is a natural process happening into the body, but now the scientists are smartly using this particular mechanism to target the proteins that are causing the problems in the body. That are causing the problems means usually we have been doing a drug discovery and inhibiting particular molecular targets. We call them as a molecular target. So here protein of interest is particularly a molecular target. So if you see into this mechanism of a PROTACs, how does the PROTACs molecules work? Basically, this will help you to understand how the PROTAC works. This particular slide. Okay, so POI is POI is a, you can see here POI is a protein of interest. That means the molecule, protein molecule which you want to target. And if you want to target that particular molecule and inhibit the concentration or expression of that molecule into the body, then you need to inhibit it or degrade it. So in this case particularly, it is being degraded. Okay, and here what happens is PROTACs are designed in such a fashion that they bring in the protein of interest closer to the E3 ligase, and then um E3 ligase do the ubiquitination of the protein of interest. Let us look into the step by step of this. First step in this one is a formation of a formation of the ternary complex. PROTACs a thermodynamic center on creating a stable connection between E3 ligase and the protein of interest, as I mentioned. This connection forms a ternary complex that serve as a foundation of the subsequent steps. And then second step is a ubiquitination process. Within the ternary complex, the protein of interest or POI undergoes ubiquitination, a process crucial for its degradation. That is kind of a tagging. Ubiquitination involves the activation of ubiquitin through ATP dependent mechanism that we have seen into the earlier slide. I introduced it to you. Activated ubiquitin is then transferred to specific cysteine residue of the E3, E2 enzyme that we are seeing. Finally, ubiquitin is donated to the protein substrate through E3 ligase complexes, facilitating the tagging of protein of interest. And then the proteasomal degradation happens. Once the ubiquitinated protein of interest is targeted for the tagged for the degradation in the proteasome. This degradation step is vital for removing the targeted protein from the cellular environment. Here, the E3 ligase specificity, the choice of E3 ligase when you are designing your PROTAC molecule is very important, because the E3 ligase will only selectively tag the protein of interest. That is what the key for designing the molecule, uh, uh, PROTAC molecules comes into the picture. The recruitment depends on the availability of a specific ligand for the recognizing the E3 ligase. Example of the ligands include those for cereblon, cellular inhibition of apoptosis protein, mouse double minute 2 homologs, and Hohenhippel-Lindau VHL. These are some of the E3 ligases. And now what happens is protein of interest need to be brought together or closer to the E3 ligase. This happens because of a specific ligands. So the ligands for this E3 ligase are thalidomide, pomalidomide, lenalidomide, and this kind of other chemical moieties which recognize or target the and they are E3 ligase, and they are called ligand E3 ligase interaction determines the selectivity and the effectiveness of the PROTAC system. In summary, PROTACs leverage the stable ternary complex to enable the targeted ubiquitination of a proteins, leading to their degradation in the proteasome. Meaning of some of the terms that we are using here, I would like to emphasize to you is that the ubiquitination is the process by which the ubiquitin molecules are attached to a protein, making it for the degradation. Proteasome, a cellular structure. You could see that here, a proteasome, a cellular structure responsible for breaking down and recycling the proteins that I introduced into. What is the E3 ligase? It is an enzyme that facilitates the transfer of ubiquitin molecule to the ubiquitin molecule to the protein of interest and tagging them for the degradation. Then E1 enzyme and E2 enzymes that we have seen into they are responsible for activating ubiquitin. Okay. This is basically the mechanism. This is a wonderful mechanism where body's its own mechanism which degrades the proteins are used to degrade the protein targeted molecular targets which are the cause of the different diseases. Okay. So in the next slide, we will see the basic strategy of the PROTACs. This is a very important slide. Like once you have understood that we are trying to utilize or capitalize the body's own mechanism for degrading the proteins using the ubiquitin dependent system, UPS system, then how to design on a molecule which will exactly work against our targeted protein in the body. So this slide basically primarily describes how it will happen. See, basically the PROTAC molecule is the has a three components as shown here. The one component that is on to the extreme left of my side is E3 ligase binder. So I told you who will tag the particular protein. It will be E3 ligase that will tag the protein. But you need to bring that protein and E3 ligase together somewhere. So this is basically, bringing them together. This PROTAC works bringing them together. So one side that binds to the E3 ligase, and there is a on extreme right, you will have the POI binder, protein of interest, or a target protein binder. So you could see that target protein binders, basically we have been designing the inhibitors. This is that kind of which binds to, but not kind of a requirement is not that it should bind like an inhibitor. It should just bring in the it doesn't need to fit into the active site and do not need to stop the kinase function or enzymatic function of that protein. It just need to bring in, bind to that protein, protein of interest or a target protein in anywhere. And there is a one crucial part, that is a linker. This linker also serves a couple of other uh, purposes than just being a linker. So we have learned that uh, basically there are three key components of the um, PROTAC design. So I hope if any questions, you people can post the questions here, and I will be happy to stop and answer those questions. Otherwise, we will go on. We basically, I will be repeating that basically we will be seeing into this one that the PROTAC has three components. One is the E3 ligase binder, which brings the E3 closer to the protein, and there is a protein binder which brings the protein closer to E3 ligase for ubiquitination. So now we will go to the design. Basically, we will see the basics. I mean, we will not be able to go into the deep of the design strategies, but we will see the basic. Here, the one that I'm going to discuss with you is that structure-based PROTAC design strategy. Basically, involves utilization of structural information often obtained through the techniques such as X-ray crystallography to guide the design and optimization of a PROTAC molecule. This approach focuses on understanding the three-dimensional arrangement of the components within the ternary complex of the PROTAC. Ternary complex, you have seen that there is a PROTAC, target protein, and E3 ligase. These three components together is called the ternary complex. At the protein-protein interaction interface. By gaining insight into the spatial orientation and interaction between the molecules within the ternary complex, researchers can optimize the design of a PROTACs to enhance their efficacy in inducing the targeted protein degradation. That is what our final goal is. Basically, structure-based PROTAC design allows us more rational and informed approach to PROTAC design by considering the structural details of a protein-protein interaction interface. This can involve optimizing the linker region. As I mentioned, that linker is connecting E3 ligase domain and the warhead group of the PROTAC to ensure the optimal positioning and interaction with the target protein and the E3 ligase. Additionally, structure-based design approach can aid in the selection of an appropriate protein binding warhead and the linker chemistries based on the structural characteristic of the targeted protein and E3 ligase. By leveraging the structural information, tools and computational approaches, this structure-based approach can enable us to refinement of a PROTAC molecules to enhance their binding affinity, selectivity, and overall efficiency in recruiting the E3 ligase to the target protein. Here you can understand that our main goal is that we want to bring in the protein of interest closer to the E3 ligase, or we want to recruit the E3 ligase to the targeted protein. Then the E3 ligase will do its work of leading to the effective ubiquitination and subsequent degradation of the target protein. This approach holds promise for the rational design and optimization of a PROTAC, potentially reducing the empirical nature of a PROTAC development and increasing the likelihood of a success in generating a potent and selective targeted protein degraders. In summary, on this slide we can see that we can harness or utilize the structure based PROTAC design approach to optimize the protein interfaces within the ternary complex, thereby improving the efficacy of a PROTAC molecule in targeted uh, protein degradation. So on to the design 2, we will see that this mark 2 and 1 degradation. This is particularly this is the enzyme that is involved into the uh, cancer, and this is a target. Here the target is a smart A2 and smart A4, and here we are able to see that there is a design, and you can see the actual. For this is the first slide where you are able to see the the linker, different linkers, and then the warhead group and the E3 ligase, actual E3 ligase ligand. Okay. So what are the different steps that are involved into designing the particular PROTAC molecule? So let us look into. First thing is that identification of a ligand binding site. In the first, we have to identify where the ligand will bind. How the structure based PROTAC design will help us is that the co-crystal structure of the particularly in this case, in a complex with the ligand revealed a K interaction at the binding site. You can see onto the slides, which for example, the ligand was observed to bind deep in a acetyl lysine binding site in forming interaction with a specific amino acid residues such as uh, Y1421 and N1464 that are marked there. These interactions were crucial for the design of a ligands with a high affinity and selectivity for this particular enzyme. So, this is an example where we can see that how the PROTAC molecules are designed using the co-crystal structure. Further comes, as usual, the optimization of the ligand interaction. The co-crystal structure of the ternary complex of the this particular enzyme provided insight into the interaction between the PROTAC molecule and the target protein. Here, what has happened? Actually, the PROTAC molecule and target protein co-crystal structure was obtained. It was observed that the large portion of the de novo protein-protein interaction exists around the fluorocyclopropylamide moiety that you can see at the terminal. This information guided the optimization of the PROTAC molecule to enhance its interaction with the target protein, thereby improving its degrading activity. The design of the linker is one of the crucial parts. There also this approach help or structure co-crystal structure help. The co-crystal structure of the ternary complex provided valuable information for the design of the linker connecting the ligands targeting VHL. This is particularly ligase and SMARCA2. The structural information revealed the importance of the linker interacting with the lipophilic phase, partly formed by the tyrosine 98 of a VHL. So this is rational design of a PROTAC. The structural information obtained from the cross-crystal structure allows for the rational design of the PROTAC molecule. Specific modifications can be made to the PROTAC molecule to optimize its interaction with the target protein and the E3 ubiquitin ligase, leading to the development of a potent and selective degraders. Okay. We have to as a remember that as the structural biologists and in silico chemists are integral part of our projects, and this is where they add a lot of value to the project. So we need to take help time to time, in this case also PROTAC. Then another side, very important aspect of the PROTAC design, we are going to see that, and that is a exit vector. The exit vector is a critical component of a PROTAC molecule, affecting their specificity, efficacy, pharmacokinetic, and overall therapeutic potential, an such an important part, that exit vector. Here it is highlighted by the piperazine ring. So what is the importance of this particular exit vector in the PROTAC molecules? Okay, here importance is that they help in achieving the specificity and selectivity. The exit vector plays a critical role in determining the specificity and selectivity of a PROTAC. A well-designed exit vector ensures that the PROTAC can effectively distinguish between the target protein and other protein. Otherwise, the selectivity will be lost, and this helps in minimizing the off-target effects. Orientation and conformation. The exit vector influences the orientation and conformation of the ternary complex formed between the PROTAC and the target protein, the E3 ligase. The spatial arrangement dictated by the exit vector is critical for the ubiquitination process, as it affects the proximity and orientation of the ubiquitin ligase to the lysine residue on the target protein that are to be ubiquitinated. So solubility and permeability is also affected or influenced by the exit vector. Its hydrophilicity or hydrophobicity can affect these parameters of a solubility and cell permeability of the PROTAC molecule that we are designing. These properties are essential for ensuring that the permeability of the PROTAC is fair enough for the development of it as a drug. See, a well-designed exit vector, this is a very critical component of the PROTAC design, prolongs its circulation time in the body and optimizes its distribution to the tissues where the target protein is located. Another aspect that we need to pay attention when we are selecting or designing the PROTAC molecule, and particularly the deploying an exit vector into the PROTAC molecule, is that the ease of synthesizing the exit vector and incorporating it into the PROTAC molecule affects the scalability and cost effectiveness of the PROTAC production. The design of the exit vector must consider synthetic accessibility to facilitate the efficient production of the PROTACs. The ternary complex dissociation. The exit vector can influence the rate of dissociation of the ternary complex, which we are going to ternary complex. We will see the significance of the ternary complex into the next slide. But that dissociation is also affected by the design of the exit vector. The slower dissociation rate may lead to more efficient target protein degradation, as the E3 ligase has more opportunity to ubiquitination of a protein of interest. Now, in the next slide, we will see the importance of the ternary complex. Okay. So the ternary complex that is formed. But let us first begin with this one. This is basically a complex formed by the interaction of the PROTAC molecules, ligase, and a protein of interest. These are the three components of this ternary complex. Okay. So understanding the stability, cooperativity, and the kinetics of the ternary complex is very crucial for optimizing the design and improving the efficacy of the PROTACs. So how does it, what are the aspects that are important? So let us look into that. The stability of the ternary complex is directly related to the efficiency of the target degradation. If the ternary complex is not stable, then ubiquitinations will not happen enough, and then the protein will not be tagged for the degradation. A more stable ternary complex can increase the probability of a successful target and degradation, leading to higher potency of the PROTAC. Also, the stability of the ternary complex affects the selectivity or improves the selectivity. Cooperativity between the PROTAC and the protein of interest and E3 ligase can affect the selectivity of the product for its intended target. A higher degree of cooperativity can increase the specificity or selectivity of the product for its target, reducing the likelihood of the off-target effects. Optimizing kinetics. The kinetics of ternary complex formation and dissociation. You are seeing into this slide that the complex formation at the top and the dissociation. Okay. So the kinetics of ternary complex formation and dissociation can affect the rate and the extent of a target degradation. By optimizing the kinetics of the ternary complex, we can improve the efficiency of a target degradation and reduce the likelihood of a premature release of a protein of interest before it can get ubiquitinated or degraded. Designing more effective PROTACs is our goal. By understanding the stability, cooperativity, and kinetics of the ternary complex, we can develop the strategies to optimize the PROTAC design and improve their efficacy in inducing the target degradation. For example, they can modify the linker between the POI binding and E3 ligase binding moieties to enhance the cooperativity and stability of the ternary complex. In summary, I can tell you that understanding the stability, cooperativity, and kinetics of the ternary complex is crucial for optimizing PROTAC design and improving their efficacy in inducing the target degradation. By studying the PROTACs, which have the potential to revolutionize the drug discovery and therapeutic interventions, I can tell you that you will be, as a drug discovery scientist, as a medicinal chemist, as a pharmaceutical, as a pharmacy graduates, you will be gaining a lot of advantage over the other people. And this is a very hot topic. I mean, the further we will look into this is all. Now I will in general discuss with you that basically we have been seeing that particular disease. This is in a very general for the students, those who are attending this. We have been we have been understanding the disease biology, and there are particular targets that cause the that are the at the basis or at the genesis of the particular disease. So what is our solution? How do we manage those proteins that are causing the, basically we have been seeing that we develop the inhibitors, and inhibitors are into the market. But now in a last couple of years, I mean, I can say specifically last 5 years, we are seeing that the couple of PROTAC molecules, okay, uh, uh, PROTAC molecules are into the clinics, and this is going to be the very important aspect of the drug design in future. It has been widely used into the oncology drug discovery as of now, but currently recently the people have also come up with the couple of PROTACs that are targeting the neurogenerative diseases and the skin diseases and even the viral diseases. All that is happening. And the advantages of the PROTACs is that particularly you have seen that in case of inhibitors, you need to target the active site of that particular target, molecular target, and then you need to design a molecule that fits into that active site and stops the enzymatic function in case of protein kinases, like a kinase function, But in this case, you don't need to be to achieve you don't need to target specifically the active site. You can target any site onto the protein of interest or a target protein, and then bring it to closer to the E3 ligase, and then it can further go. Another this one, those where the were not a druggable targets, they are also can be targeted by this strategy of the PROTAC molecules. Another aspect is that particularly enzyme inhibitors only inhibit the enzymatic activity of the particular protein, but other functional roles that the particular protein plays into the body cannot be stopped just by inhibiting or just by blocking the active site of that particular targeted that particular molecular target or a protein of interest, or which is the protein which is causing the disease. So in this case, you are completely degrading the protein, so there is a very little chances of getting the mm, achieving the resistance, or also by any other means if that performs the function that is not possible in case that is degraded. I will take I'll take you through the kind of a metajivan, how it works. Metajivan Research Consortium is an initiative of a Crop Jivan Agro Research and Development Private Limited. Particularly, Metajivan Research Consortium works in the pharma, drug discovery, and biotech domain. How does it happen? This is a consortium of a couple of companies and academic institutions like yours. Here, what we do is we cater to the needs of each other. This is a win-win situation. Suppose there is any company that is specialized into, as here you can see that company 1 is a chemistry company, company 2 is a biology company, company 3 is a clinical, and company 4 is manufacturing. And these all they come together and they form a consortium, and we cater to the needs of each others and also the needs of our customers. So we have wide range of companies and institutions associated with us. So if there is any project that want to be run by the suppose there is a company 1 and that is doing a chemistry and synthesizing some molecules, but that molecule need to be evaluated for the biological activity, then company 2 will do that job for them, and they collaboratively can work onto the MetaJivan platform and get their projects ahead and do the work. Because one company, drug discovery is a highly interdisciplinary subject. One company cannot deliver everything. At least the smaller companies cannot deliver, and there are a lot of smaller companies in India. So we have formed this consortium where we cater to the different needs, and also within the consortium, we help the member companies each other to solve their problems or to cater their needs. How on other aspect that we collectively serve the customer or the clients. If the client or the institution has some requirement, they want some project to be run, and that project involves the chemistry also, biology also, or the manufacturing, GLP, GMP formulations, that we get it done through our companies on a one platform. That is what the advantage of coming together and doing together the research. Because drug discovery or the biotech research are highly interdisciplinary research. Most of the smaller companies could not deliver single-handedly. So that's all for me, and if there is any questions, they are welcome. So I will like to also bring into your notice that I'm very active onto the LinkedIn. If you want all this information about the PROTACs, their strategies, and a lot of other examples, those are into the clinics. That also can be seen into my, you can search Pursuhottam Dewang on LinkedIn and follow me. There you will get a lot of information about the PROTACs and other drug discovery strategies. If there is any question that I will answer, otherwise I'm done.
Thank you, sir. Delegates having question can type your question in. One question is there, sir. Is there any approved PROTACs by FDA? So your voice is not audible.
So you're not audible.
So thank you very much, Hitesh. The PROTACs as of now are not approved, but they are into the advanced stages in a clinical trials. So basically, PROTAC is a very recent development into drug discovery or a strategy, and the success that people have achieved by advancing them into the um, clinical trials has been celebrated. And as you have seen from my first slide that I have shown, huge amount of money being deployed into this field. Any other question?
Delegates can type your question in chat box. We will wait for some question, sir, for some time.
No problem.
So we have question: is PROTAC follow the Lipinski's rule for 5?
No. Very good question. If you see the structure of the PROTACs, as you have seen, one of the very good question. Actually, PROTAC is a topic which can be discussed, in as a series itself. Okay, very good question. PROTAC do not follow the rule of 5, and that was the inhibition initially people had. Whether this kind of a molecule, such a huge molecule, can be made into the drugs or can be seen them as a probable drugs or not. But now that inhibition has gone, and lot of molecules, people are developing the molecules for the even for the oral administrations. I mean, it was a challenge that how come this much big molecules will have the cell permeability, solubility, and all those aspects that we see basically for the molecules below the 500 molecular weight. But PROTACs do not follow. Very good question, Hitesh.
Okay, I think no more questions are there.
Thank you very much.
Thank you so much, sir. We highly appreciate your valuable guidance on. You highlighted on mechanism of PROTACs, various design strategies of PROTACs. You explained the importance of ternary complex very well. Thank you, sir. Thank you so much. Now I request you to please accept the certificate as a token of love on behalf of our college.
Thank you very much, madam. Thank you.
Thank you. Thank you once again. Thank you so much.
Thank you. I am leaving the studio. Okay, bye.
Yes, sir. Yes, sir.